In this lecture, we will examine together humoral regulation of blood pressure. The primary resource for this lecture can be found in Cardiovascular Physiology Concepts, published by Walter Kluers in the 3rd edition in 2021. So let's begin by looking at circulating catecholamine. There are two principal catecholamines that are involved with cardiovascular regulation, and these are norepinephrine and epinephrine. The primary source for norepinephrine is sympathetic efferent nerves. When these nerves are activated, they release norepinephrine and a small fraction diffuses into the blood. A secondary source for norepinephrine will be the catecholamines that are released from the adrenal medulla. The second catecholamine is epinephrine, and that is primarily formed and released by the adrenal glands, the adrenal medulla. In this case, we find that the adrenal glands release catecholamines in the ratio of about 80% epi and about 20% norepinephrine. In both cases, whether it's neuronal release or from the adrenal glands, it is stimulated by sympathetic activation. Now let's look at the formation and release of these two different catecholamines by sympathetic nerves and the adrenal medulla. There are common pathways, and so I'm only showing one figure here for both of these sources of catecholamines. The catecholamines originate from tyrosine that is transported into a neuron or into an adrenal chromaffin cell. Once this tyrosine is in the cytoplasm, it can then be converted by tyrosine hydroxylase to L-DOPA, and then DOPA can be decarboxylated to dopaamine. Dopaamine then can be transported into neuronal vesicles, as shown here, acted upon by an enzyme called dopamine beta hydroxylase to form norepinephrine. And in the adrenal glands, dopamine, like in the neuron, is transported into the chromaffin granule and is also acted upon by dopamine beta hydroxylase to form norepinephrine. Now, some of this norepinephrine is packaged for subsequent release by these granules, but additional norepinephrine is transported out of the granule into the cytoplasm, where it's acted upon by another enzyme that causes the methylation of norepinephrine, and the addition of that methyl group transforms it into epinephrine. And epinephrine then is transported back into the chromaffin granule, where it is stored for subsequent release. So when sympathetic activation occurs, either within the nerve itself or sympathetic nerves which innervate the chromaffin cells within the adrenal medulla, that activation by sympathetic nerves causes the release of stored norepinephrine in either the neuronal vesicle or in the chromaffin granule, or cause the release of stored epinephrine. Now let's look at the metabolism of these catecholamines. If we look at both norepinephrine up here and epinephrine, there are two types of enzymes that can act upon these catecholamines. One is monoamine oxidase, which will lead to the formation of dihydroxymandelic acid. And then that is acted upon by catecholamine O-methyltransferase, or COMT. And the epinephrine, instead of being acted upon by male, can be acted upon by COMT, and so can norepinephrine. And they will then form these particular products, norepinephrine or metanephrine. Now, either of these products can be acted upon by MAO to form benylyl-mandelic acid, which we often abbreviate VMA. Now, these are important metabolites because they are used to assay for abnormally elevated production of catecholamines in the body. Let's look at what receptors these catecholamines act upon. In this slide, I want to be very, very clear about something. When we talk about norepinephrine, we're binding to both beta and alpha receptors. Now, in heart muscle and nodal tissue of the heart, this is the response. I'm using the word response. It's not receptor affinity or binding to the receptor or the number of receptors. It is the overall response. So the order that I am showing here for you might be different than what you might see if you first just go to a pharmacology textbook and you look at how norepinephrine interacts with receptors, because they might be talking about the pharmacodynamics of its interaction. And I'm looking at the overall response because physiologically and clinically, this is what we are looking for. So in terms of norepinephrine, the response in the heart, such as increased heart rate, contractility, is largely a beta-1 receptor mediated response, secondarily a beta-2, as I talked about yesterday. There is a small alpha-1 response, but it's very, very minor compared to the beta-adrenergic response. In blood vessels, the response is primarily mediated by alpha-1 receptors and secondarily by alpha-2 receptors on the vascular smooth muscle. And then there is also some binding to beta-2 receptors, although it's rather weak. And so the response to beta-2 receptor activation is completely masked by the overwhelming alpha-1 and alpha-2 response in the blood vessel, which produces contraction, and this response, beta-2, produces relaxation. Now let's look at epinephrine. Epinephrine also binds to beta-1 and 2 and alpha-1 and 2 receptors. Now in the heart muscle and nodal tissue, the response is beta-1 primarily, beta-2 secondarily. In blood vessels, epinephrine, the response to normal small increases in epinephrine or moderate increases in epinephrine, the response is primarily a beta-2 response, a vasodilatory response. And then it will also respond in part to, via alpha-1 and alpha-2 adrenoceptors. But I want you to note here, as we're going to see in a subsequent slide, that the response to epinephrine in blood vessels is highly concentration dependent. And my little note down here is also an important note. The response depends upon the available number of receptors, ligand, receptor affinity, and downstream signal transduction coupling. That's why I present this as the overall response rather than trying to dissect out the detailed pharmacology and how it interacts with receptors and signal transduction mechanisms. Well, let's see what happens if we inject a low dose of epinephrine into the body versus norepinephrine. If we inject a low dose of norepinephrine into the body at this point, now this is arterial pressure, systolic pressure here, diastolic here. We will notice that the mean pressure really doesn't change very much, although we get an increase in systolic and a decrease in diastolic. This is primarily due to the fact that we get a large increase in stroke volume in response to the epinephrine. But we don't get much of a change in mean arterial pressure. That means that although cardiac output is likely increasing a significant amount because heart rate is increasing, we're not getting a change in mean pressure, which suggests, and indeed this is what occurs, we're getting systemic vasodilation. And that's why the mean pressure doesn't change, although we see that cardiac output, or in this case, heart rate, is increasing. Now, in contrast, norepinephrine, which structurally only differs from a single methyl group on the molecule, has a very different type of response. When you inject it, mean pressure does rise. The pulse pressure also rises. We see initially there might be a small increase in heart rate, and then heart rate declines. Now, why would heart rate decline here if this is a drug that is a beta agonist and should be increasing heart rate? The reason why heart rate falls is the activation of a barrel reflex. But let's concentrate a little bit more on epinephrine because in the last slide, we were looking at a low dose. And I want us to contrast a low dose of epinephrine versus a high dose of epinephrine. With a low dose of epinephrine, we will see a large reduction in systemic vascular resistance. So that would tend to lower blood pressure. But at the same time, with that low dose, we see a large increase in heart rate and a large increase in cardiac output. Now, arterial pressure doesn't change very much. In fact, it declines a small amount because the change in SVR is greater than the change in cardiac output. Now, it doesn't look that way on the slide here because I am showing this slide as percent change in cardiac output. And a 50% decrease in SVR would be equal to a 100% increase in cardiac output. Then mean arterial pressure would not change, because when you multiply, mean arterial pressure is approximately SVR times cardiac output. You can't use percentages when you multiply. You have to use factors like a 1.2 or a 1.4 or something like that, or a 0.5 change. You can't use percentages. But that's the response to a low dose. On the other hand, if we look at a high dose, we say, wow, we give a high dose of epinephrine, we don't get this fall in SVR. SVR might not change at all. It might increase a little bit too, but it just doesn't change very much. And the reason for that is in contrast to this, which is a beta receptor mediated response, at the high dose, we're also bringing in its effects on alpha-1 and alpha-2 receptors, which are vasoconstrictors. So now we have two mechanisms operating, a vasodilatory mechanism and a vasoconstrictor mechanism, and they kind of cancel each other out. So what you need to keep in mind here is that vascular beta-2 receptors have a higher affinity. Now we're talking about pure pharmacology here. These receptors bind epinephrine more avidly at a lower concentration than the alpha-1 receptor. So there's a difference in affinity for epinephrine, and that explains the difference in vascular responses between the low and high dose. Now, with the high dose, we are getting a pressure response. Arterial pressure will rise, not because SVR is increasing, but because cardiac output is still increasing. And heart rate is increasing, which contributes to the increase in cardiac output. Now, it is interesting here, you ought to note this, that despite having a higher dose, what happens to heart rate? It's not as high. It does not increase as much here with the high dose as it did there. Well, why not? In both cases, we're acting upon beta receptors in the heart. Well, in this case, with a high dose, we're getting an increase in blood pressure. And that increase in blood pressure, which did not occur here, triggers a baroreflex, which activates the vagus nerve to the heart. And that partially attenuates the beta receptor effect on the heart, which would tend to increase heart rate. But now in the presence of vagal activation, it suppresses that or attenuates that increase in heart rate. So a key concept here is that baroreceptor reflexes modify cardiovascular responses to hormones and drugs. And so even though you will learn about certain drugs that will have certain effects on receptors in the body, once you inject that drug into the person, you are now going to have that response modified by baroreceptor reflexes, because if that drug affects arterial pressure, either increases it or decreases arterial pressure, you are going to produce a baroreflex response that will modify the actual drug actions. So circulating catecholamines are elevated by what conditions? Well, anything that increases sympathetic efferent nerve activity, because the sympathetics do release norepinephrine, but they also trigger the adrenals to release catecholamines. So just like we saw yesterday with sympathetic activation, it would be increased physical activity, stress, heart failure, or shock conditions. Well, we can add a new one to the list here, and that is the condition of a pheochromocytoma, which within the adrenal medulla would cause this episodic release of catecholamines with a lot of cardiovascular effects. Now we're going to look at another system. This is an extremely important system. It's a much more complex than the catecholamine system, but it is one that we take advantage of in treating a lot of cardiovascular disease. There are tens of millions of Americans that are taking drugs that block the renin-angiotensin-aldosterone system because it's used in the treatment of hypertension and it's used in the treatment of heart failure. So this is a very important system to understand. Now we're going to begin just by looking at what triggers the formation of angiotensin II. So in this little model here, I am showing the kidney and sympathetic activation of the kidneys operating through beta-1 adrenal receptors in the kidney will lead to the formation and release of renin by the kidneys. Or hypotension, systemic arterial hypotension, or a reduced blood pressure in the afferent arterial of the kidney, which can be caused by renal artery stenosis. That will lead to a production of renin and release, or decreased sodium delivery to the distal tubule. But these are the primary mechanisms that cause release of renin by the kidneys. Renin then is a proteolytic enzyme that acts on a substrate produced by the liver called angiotensinogen. And renin then will cleave off peptides to form the decapeptide angiotensin I. And then angiotensin I is acted upon by angiotensin converting enzyme, or ACE. Knocks off two more peptides to form angiotensin II. Now what I'd like to do is to go through the detailed actions of angiotensin II on the following slides. So first of all, angiotensin II leads to an increase in blood volume, which I have highlighted here. And it does it by a variety of mechanisms. One is that angiotensin II will stimulate the kidneys directly to enhance sodium retention, which causes fluid retention and leads to an increase in blood volume. Angiotensin II also stimulates the adrenal glands, but now the cortex, not the medulla. The medulla associated with catecholamines, the cortex associated with mineralocorticoids such as aldosterone. So it stimulates aldosterone release, which acts upon the kidneys, also leads to sodium retention and fluid retention to increase blood volume. It also stimulates thirst centrally up in the hypothalamus, which will cause a person to drink more fluids to increase blood volume. And it also stimulates vasopressin, or antidiuretic hormone, which is produced up in the hypothalamus and then released by the pituitary. Other actions include angiotensin II increases systemic vascular resistance directly because A2 is a powerful constrictor that acts upon a receptor that we call the AT1, or the angiotensin type 1 receptor, which is located throughout the vasculature, in many regions of the body, in fact. And in the vasculature, it causes vasoconstriction. Angiotensin II also augments or enhances sympathetic activity. And it does so by enhancing norepinephrine release, acting centrally up in the brain, but also peripherally on the nerves. It enhances norepinephrine release and inhibits norepinephrine reuptake by the sympathetic nerve terminals through prejunction of AT1 receptors. By inhibiting that reuptake, then you have more norepinephrine in the intracellular cleft for any given level of sympathetic activity. So in that way, it enhances or augments sympathetic activity. It also stimulates cardiac and vascular remodeling. And so we give angiotensin II inhibitors, or drugs that block this RAAS system, in order to attenuate the remodeling that might occur in the heart in response to heart failure or following myocardial infarction. So as a key concept here to summarize this, angiotensin II increases arterial pressure by acting directly or indirectly on the kidneys, the vasculature, and the heart. Now, let me just follow up a little bit more about aldosterone effects before I leave the RAAS system entirely. When you have aldosterone acting on the kidney, it acts in the late distal tubule of the kidney. And what it does, it stimulates the uptake of sodium from the urine, but it is in exchange for potassium and hydrogen ion. So when you have increased release of aldosterone, yes, it helps to reabsorb sodium and water, which follows the sodium. So it helps increase the blood volume. But at the same time, it causes an increased loss of potassium. And so therefore, high levels of circulating aldosterone are associated with increased potassium loss to the urine and therefore hypokalemia. Now let's go on to another humoral agent, and that will be the natriuretic peptides. The first natriuretic peptide that was discovered that had a lot of biological activity and significance was what we call atrial natriuretic peptide, or abbreviated NANP. It is a 28 amino acid peptide synthesized by the heart, by the atria within the heart, primarily by the atria. So the heart is not only a pump in the body, it's also an endocrine organ. It synthesizes a very important hormone, ANP. And its release from the atria is stimulated by atrial stretch, sympathetic stimulation, or increased circulating levels of angiotensin-2 or endothelin-1. And so, especially these first two here, but even the third one because of angiotensin-2, this is what occurs with heart failure, for example. In heart failure, blood backs up into the atria because the ventricles can't pump it out as well. And you get atrial stretch, and it stimulates the production of ANP. There's another natriuretic peptide that we call brain natriuretic peptide, because we call it brain or BNP because it was first discovered in the brain. Although what our focus is on the fact that it's also produced by the heart. It's produced in the atria and in the ventricles. It's a little larger amino acid peptide than atrial natriuretic peptide. It's produced by both chambers of the heart, upper and lower chambers of the heart, but because of the much larger ventricular mass, most of the release into the blood is from ventricular tissue. And its release is stimulated by ventricular dilation, as occurs in many forms of heart failure, as well as by sympathetic activation, angiotensin-2 and endothelin-1. So BNP and AMP, as we will see, have very similar actions and are stimulated in a very similar manner, although one is coming more from the ventricles, and the other one, AMP, is coming more from the atria. Now let's just look at AMP here, but I could have substituted in BNP here in this diagram. So I'm showing here in this diagram that atrial distension, sympathetic activation, A2 or ET1, will cause the heart to release AMP. Now, what does AMP do? It acts upon the kidney to increase glomerular filtration rate. So it stimulates GFR. It also then inhibits renin release. If you inhibit renin release, you decrease circulating levels of angiotensin-2 and aldosterone. And by doing that, you will produce natriuresis, which is sodium loss. You'll promote sodium loss by the kidneys and also have a water loss or diuresis. So it acts as a diuretic. That will decrease blood volume. If you decrease the blood volume, you will decrease central venous pressure, which will decrease cardiac output, which will decrease arterial pressure. ANP also will act directly upon the veins to increase their compliance. In other words, relax the smooth muscle within the veins, which will cause a decrease in central venous pressure. And then ANP acts on arterioles to dilate them and to decrease SVR, which will lead to the decrease in arterial pressure. So if you think about this diagram and what changes are occurring, it appears as though what AMP is doing is just the opposite of what RAAS activation does. And that's the easy way for you to remember what the actions of natriuretic peptides are. So as kind of a key summary for the RAAS system, for natriuretic peptides, they counterregulate the renin-angiotensin-aldosterone system. And a little clinical aside here is that measurement of blood BNP or its precursors is an important tool in the diagnosis of heart failure. Now let's look at the hormone vasopressin. Vasopressin has other names. We call it arginine vasopressin or AVP. We call it ADH. That's antidiuretic hormone. It's all the same hormone. Vasopressin is synthesized in the hypothalamus, in the lower part of the brain, but above the brainstem. Normally, and by far the most important factor stimulating it is hyperosmolality, because if you become dehydrated, you become hyperosmolar, your blood, and so that is sensed by the hypothalamus, which will then form antidiuretic hormone that's released. It acts on the kidneys and helps to retain fluid, to restore fluid balance and electrolyte balance in the body. Well, that's the most important, but there's other factors that stimulate the production of vasopressin. The other important factor is decreased atrial receptor firing. There's a type of receptor, particularly located in the right atrium, what we call a type A receptor, that its normal firing through changes in the pulsatility from the right atrium, that pulsatility in the firing of those receptors inhibits hypothalamic production of AVP. But if you have, let's say, a hemorrhage condition where these receptors will show decreased firing, you remove that inhibition on the hypothalamus, and so you have increased production of vasopressin. Angiotensin II stimulates it, acting directly on the hypothalamus through AT1 receptors, and so does sympathetic stimulation. That vasopressin is formed in the hypothalamus, travels down to the pituitary gland via axons, and from where it's released into the blood. Vasopressin acts on receptors, on blood vessels, V1 receptors, that cause constriction, which increases SVR, which increases arterial pressure. In the kidneys, vasopressin acts on V2 receptors, leading to fluid reabsorption, increased blood volume, and increased blood pressure. Now, normally the primary action of vasopressin is the renal effect. However, in conditions of shock, where we do get a large release of vasopressin, its concentration becomes high enough where it indeed does contribute to the increase in SVR through the V1 receptors. Now, the renal actions of vasopressin, it acts primarily in the collecting duct, and ADH stimulates the reabsorption of water. So we've looked at quite a bit in this lecture, and I will take the time just to go through these salient features of the lecture. Circulating norepinephrine and epi, originating from sympathetic nerves in the adrenal medulla, alter cardiac and vascular function through alpha and beta adrenoceptor coupled pathways. Both of these hormones act on both types of receptors, as we learned, but in the case of epinephrine, there is a concentration dependency in terms of its vascular response. Renin released by the kidney stimulates angiotensin II and aldosterone formation, which act on the kidneys, the vasculature, and the heart to increase blood pressure. Natriuretic peptides that are released by the heart counter-regulate the RAAS system. And then vasopressin acts on blood vessels and the kidneys to increase arterial pressure.